Good morning, everybody. We have a surprise for you in all languages, thanks to Valérie.
Hello, hola, hallo, dzień dobry, Dani Bona, molo, guten Morgen, doloro jutro.
We're doing our best here with all languages.
Buenos dias, buongiorno, et pour finir en bon normand, bonjour.
We are very proud and happy and also deeply moved to host you here with Valérie. This is the second installment of this day dedicated to clinicians, family, patients, dedicated to patients and families. There are close to 200 people on this day dedicated to MYT1L gene. We have a beautiful program organized by Valérie. We are very fortunate to also have international researchers, but also French researchers that are involved in research on MYT1L. This is also organized by the French branch on rare diseases, and we'd like to thank them for their support throughout the years and months. Thanks to the interpreters who are going to translate live the 9 presentations. Thank you very much. Thank you for being here. Thank you for traveling all the way. To seize this opportunity to connect and really spend a very good time here. Well, I think there's nothing more I have to say, but I'm going to start and sing. Happy birthday to you. Happy birthday to you. Well, the interpreter is not going to sing any further, but happy birthday to the extra brave, Extra-Vaillants being the name of this association. So heartfelt happy birthday. Yeah, this is a very nice opportunity. 4 days ago, almost to the date, we were filing the paperwork and created this association. So it's really today that we're celebrating the 4th anniversary. It was created the association based on this diagnosis, also all those families that we met, and also all professionals that decided to come together and do whatever they could here in this very city of Rouen. So I suppose all the stars were aligned for us to create this association. So you have the logo, which was drawn by my little sister. In the meantime, it's changed slightly, but this is how it looked like 4 years ago. The first project we had as an association can be summarized with those 4 words. First, we wanted the disease to be recognized. You know, it's important from an administrative standpoint to be recognized. And it's also important for parents to be heard, to understand and to know that they are understood and recognized, that they're not mad. Also, the second objective was to really deepen the underlying mechanisms at play and also better understand what are the impacts of this disease. We also wanted to inform families, obviously, but also the medical professionals at large, medical doctors, but also nurses, healthcare professionals, inform them of the creation of our association and provide them with as many information as possible, and help connect, create links and bridges, so connect families, but also the different players in all areas that work around our association on the daily, or if not on the daily, that are here to really support us. Now, a quick timeline and retrospective of what we've done in 3.5 years. Obviously, we filed for the creation in June of 2021, but everything really started in November 2021 when we published the research study by Dr. Coursimault and her colleagues. This is also in November that we launched our internet website. And so this was really the date when we really helped people connect and help them find information. In 2022, we organized the first day. There were a few less people, or a lot less people in the room, and it makes the world to me to see that today there are people from 10 different countries of the world. We have 50 children here today in the room. We have 34 members of the Vaillants Association here. So that's great. And thank you for that. Also, in 2022, with Professor Poitou and Dubin, we also started a study with the aim to better understanding eating disorders associated to this. We also started to work with international researchers, and those researchers will give a talk later in the day. And I think it's very important that we were able to create those connections beyond borders. And it started in 2022, and then we were able to connect on the other side of the Atlantic as well. Obviously, we also wanted to get this disease recognized as an orphan disease. We filed for recognition in 2021, and we were successful in February of 2023. You now have the French version, English translation, and Spanish translation of this definition. We also received funding to create a framework or a protocol for treatment. In this framework, we describe the disease in details, and also we describe recommendations, guidelines, best practices. This is for healthcare professionals, GPs as well, with a number of data sheets, etc. And we know it's very important the first time you go to your GP to have that in your armamentarium. We also set up a project to get guidelines for the different associations and the different entities that work around rare diseases, so French entities. And again, we have a cheat sheet, if you will, on everything that has to do with this disease. That's very important because, as you know, the diagnosis is rather late in life. Median age is 11. It's better now than it used to be, but nonetheless still rather late in the child's life. And so these documents that we created are here to really help parents but also families to better understand what they've been through and help them have a good description of all that it takes. So on that cheat sheet or flashcard, we are going to create flashcards to make things simpler and easier to use, especially with and for children. And everything is on our website, so I really try and focus on the most important things.
Now, in 2024, what's really important is that we've connected with the American teams, and I'll go over the role that's been played by Elizabeth Wood. I'll be talking about that a little bit later, but I just want to say that the families, all of the actions taking place are not restricted to France. We have connections that go way beyond and that have been around for a while now, even though they are taking longer to take shape. These are links, these are strong ties that we've had for a long time, and they're really important to us. And thanks to those ties, we can also connect the different medical teams and host with great pleasure, the professors that we have today.
Et 2025.
And 2025, ta-da! Here we all are. And that is amazing. It's so fantastic to have you here, having this second international conference. And there'll be another really great event taking place in Germany after, at the end of June, with Dr. Mall and his teams. And with Dr. Coursimault, we're going to run a symposium in Heidelberg. It's going to be a big international meeting. So that was a little just calendar of events and showing you what we've accomplished. Now, this is to show you everything that has happened that we can't talk about all the time. I mean, this is invisible work, collaboration between teams, working together on trying to understand this disorder, trying to help people and provide support. So there's the medical world, the paramedical world, there's the associations, patient associations, these cooperation platforms for families. These are families where families get in touch with each other, share information that can then be processed and provide information for the medical world and families in general. So this afternoon you will also hear from an organization called La Ruche des Possibles. This is a paramedical organization. The Alliance des Maladies Rares, a rare disease alliance, that's also very important because thanks to them we are connecting doctors and making discoveries. For instance, Professor Chaumet's team, which is in touch with Professor Poitou and their team. There are things that we cannot know. It's just huge, tentacular, and so there are things that are beyond current control, and these are really important, these ties. And of course, all of the doctors, I mean, I would just like you to take a look at everything that they have accomplished, everything that they've really tried to provide for you to help you. So that's what they're here for, and that's what they've done. Now, these are the tools that we've worked on and developed on in the association. But on our website, there are over 30 countries connecting, and we've had more than 9,000 sessions over the last 3 years. So it proves that what we're doing is really of capital importance, and it feeds into the whole world. I mean, it goes way beyond France. These are very important ties. These are tools that are in French and English. And we have the FALC that we've talked about and the cards or the maps that we've talked about. I think they're cards that will be available soon. Now, these are our magical links, okay? These are all the little actions and meetings and help and support that we've done and that we provide. It was so fantastic to see the kids yesterday at Happy City, seeing all those fantastic smiles and laughs. I mean, those moments are magical, aren't they? And most of them are not necessarily, live, but online. I mean, we also share Halloween events, Christmas events online. There are so many ways of connecting and sharing things together. I'd also like to thank you for your testimonies because I know how hard this is and how hard it is to talk about what you're living through. You need to accept, you need to have trust, and you need to dare to do it. And you're doing that more and more. So thank you, because it's like a gift to all of the other families. You are allowing everybody to feel reassured, to realize that there are success stories, that it is tough, but that there is a way forward. And you're reaching out to those who need it. So thank you again for your testimonies, and please keep them coming.
Là, c'est les mains tendues.
Now, this is what we call reaching out or holding out your hand, les mains tendues, that's literally what it means. This is great. We ran this event with a bank, the support of a bank, La Caisse d'Épargne, so different people looked at organizing a day with manual kind of events and activities, and they made that day really, really amazing. It was a fantastic day, so thank you. All of the young people got together in a pedagogical event, and your kids were being helped by all kinds of organizations to do all of these great fun things. So people from the medical side, the medical professions, people who are used to working in this environment. We have also companies that are funding this, helping us fund this, and they do that in different ways. And we have obtained funds to help with projects, providing iPads, for instance, for these kids, because computers are tricky for these kids. So iPads can really help them to learn and to overcome their communication disorders. And we've had a lot of people involved in all of these things. And the young lady you see there dressed in blue, you saw her yesterday actually. She's from Groupama, the insurance company, and she'll be here today as well. She's here to look after the children, to give them a snack, to run special evening events. I mean, there's so many people involved wanting to do so many things. And we've all got all the regional economic actors as well. And let me not forget, last but not least, an unbelievable support from the pupils at Donfront School. They raised 9,000 euros. So thanks to them, you are all here together in a hotel at this conference, and I certainly hope that it's going to be comfortable and interesting for you. So thank you. Thank you to Sandra and her family who worked so hard to make that happen and who ran that race. It was really, really great. Now, right from the start, we've been comparing our association to an airline. So it's the Extra-Vaillants airlines, the super valiant airlines. So I'd like everybody to stand up so that we can thank them properly. Please, a round of applause. Now, I was talking about the important ties that exist, but that are not necessarily formal. They're not necessarily organizations, but they are there and have been around for a long time. And this year, we're really lucky to see a foundation set up in the US. And we've got two people from that foundation who are here today with us. It's called the MYT1L project. Elizabeth and Kathy, could you raise your hand so we can see who you are, or maybe even stand up? We're really, really happy to have you here. Thank you. And in 2024, there will be a family day in October. But there's important work being done by the families, and I would like to thank them. I know that they're watching on remotely on screen. I know that, for instance, in Spain, Judith is really working hard to get everyone on board. In Brazil, there's a WhatsApp group with Rosie and Cassien. And in Germany, I'll be meeting the families soon. And there was a first family day in October last year. It was amazing. And we will be thrilled to meet them in June when we go to Heidelberg. So those ties are there. Things are moving. Things are happening. We are moving forward. On all of these associations, in all of these associations. So that's great. Now, there's still a lot of ground to cover, and we need to improve those ties in more countries with more actors, whether it's health professionals or associations. We have a new project for the associations in France between the Extra-Vaillants Association and the MYT1L, and I certainly hope that you will agree to help us brainstorm about that and see how we can move forward, because thanks to all of you, we will be able to move forward. I began in the first year, I felt like I was just flying a small plane. We moved on to a medium-sized plane, and now we're on an airline, a really big airline plane. But we can't do anything if we don't have a team running it. We have a pilot, but we need a whole team. We need to structure the association differently, and we need to find ways of sharing the burden and all working in the same direction. That's why I put, I need a co-pilot. And this is what I want to avoid, right? Of course. So I think that together we can find solutions. I'm sure we can. Sometimes it's tough if you have busy lives and you have children with disorders, but we can try and raise funds, and we can also outsource some tasks maybe. For instance, take somebody on to help us, recruit somebody. We could do that, but we need to make that decision collectively, and we shouldn't just leave it down to a few to run the whole thing, okay? Now, we are, of course, trying to change those challenges into brighter futures for our children. Okay, so let's begin now with our first subject. So it's about discovering and seeing where we stand at the discovery of the gene. This work is taking place in the hospital. We've split this into 3 pillars. The first part is about genetics and the MYT1L gene. Then we'll be talking about the phenotypes associated with this disorder. And then a third pillar on the clinical research of the disorder. Now, the syndrome. Let's go over the genetics of this. Each person is made up of 70 billion cells, roughly. And in the nucleus of those cells, you have the DNA, or the genome, which is really our genetic makeup. So it's like a recipe book that tells the body how to function. This DNA is unique to each person and made up of 46 chromosomes, and we'll be talking about those later. This DNA is made up of 4 letters, the nucleotides A, T, C, and G, that go in pairs. And together in the genome, there are 3 billion pairs of letters. These nucleotides, when you bring them together, make up genes, and they are like recipes. We have roughly 20,000 recipes in our genetic codes, 20,000 genes. Now, these genes are made up of different parts. We have the introns and the exons. You've probably heard of the exome, which is all of the exons that are free, and that exome makes up 1% only of the genome. It's only a tiny part of our genetic makeup, our genetic code, and yet it's in this part that most of the modifications or variations appear that are the beginning of all of these pathologies. Now, the role of these genes, or part of their role, is to make proteins so that the organism can function. And these proteins have different roles. They make the heart work, they give indications on the weight, on the size, get the bones to work. It's a little bit like a kind of telling instructions for the proteins on telling the body how to work. Now, the link between 2 genomes is identical to 99%, but the difference is called a variation. Before we used to call it a mutation, but now we call it a variation, and that means that everybody is unique and different. Now, this percentage represents 45 million variations in relation to the basic genome. And these variations, for the most part, have no impact, or can generate different eye colors, hair colors, size, etc. So only very rarely can these variations generate a disorder, a cell disorder or dysfunction, when they impact critical parts of the genetic code and generate genetic disorders. These modifications can happen in 2 different places, either on the chromosome. So you can see a missing part of a chromosome, a deletion, or in the nucleotides, in the genes, there can be modifications of the letters, either extra letters or letters changing, or some that have been deleted. For the most part, these variations are inherited from our parents. But we do have 50 to 100 new variations in relation to your parents. And that's normal, that's natural, if you like, these variations in relation to our parents' genes. And they mostly don't have any impact and don't generate any pathologies. But sometimes, very rarely, they do. And most of the time, it's a problem with the cell generation. That either happens when the sperm is being made, when the egg cells are being made, or when the divisions of the embryo in the chromosomes happen right at the beginning. The MYT1L disorder is on chromosome 2, as you know, right at the end of chromosome number 2. Now, this gene has a role. It produces a protein with the same name, the MYT1L protein, and that goes through various different stages. This protein has been modelized by the American team in 3D, and you can see it here on screen. It's made up of different parts represented by different colors here. One of the parts that's very important is the blue parts, these 6 different parts, the zinc fingers, if you like, and they produce a pocket which attaches itself to different parts of the genetic code to generate the role of the MYT1L gene. Now, this is a 2D representation of that protein. And we can see that the zinc finger is part of that 2D protein. And we're going to focus on the second and third domains or fields of the zinc fingers because they are the ones that are impacted in the disorder or the syndrome that we're dealing with. I won't go too much into detail on the role of the MYT1L gene because the American and German teams will be doing that. But I want you to see it as a conductor. It's like a conductor of an orchestra. It's a gene that's going to be conducting hundreds of genes. And the main task is to turn immature neurons into mature neurons. So that gene is going to, thanks to its protein, connect to different parts of the genetic code and play that role, turn that immature neuron into a mature one. So be careful of the name of the gene. You mustn't get it confused with the myelin production because it has no role in that process. That's what surrounds the neurons. And so MYT1 is totally different. And it's not the MYT1 gene. So they are 2 different things to not confuse. Now, we all have 2 copies of the MYT1L gene, one from our father, one from our mother. You need 2 copies, actually, so that the functions can be respected. Now, in the MYT1L disorder, there's a problem with one of the copies, a dysfunction. So only one copy will actually be a functioning one.
Ça va revenir. Parfait. Et on ne peut pas enlever la petite barre? Non?
Can we take the little bar off? No?
Donc, dans ce syndrome, on va avoir une
Okay.
seule copie qui va être fonctionnelle.
So,
Ça, c'est un
with this
mécanisme
disorder,
particulier qu'on
only
appelle
one copy
haploïnsuffisance.
is
Ça veut dire
functioning,
que
and that's a specific
la seule copie
mechanism
qui reste fonctionnelle
called
ne suffit pas à
haploinsufficience.
exercer
The
tout
only
ce rôle
functioning
attendu
copy is
théoriquement.
not enough to fulfill its role in theory.
Ça ne marche plus.
Now, this disorder is transmitted in a dominant autosomic way. That means it affects girls as much as boys, and dominant means that only one modified copy is enough to generate symptoms and the pathology. In most of the genetic code modifications are de novo, only on that particular individual, not on the parent's code, but transmission onto their descendants is a 50% one. Either the patient transmits the modified copy or another copy. Now, as far as the mechanisms go, there are two different molecular mechanisms. Either there's deletion of the chromosome, the missing part, so the chromosome deletion either takes part of the MYT1L gene away, a fragment, or the whole gene. The second mechanism at play are punctual or random variations, so missing letters. We mentioned that earlier. And as far as these variations go, these genetic code modifications, there are two different ones. But the false ones and the truncated ones. The false ones is an amino acid that's abnormal. So the protein will have a specific form because there is a random problem. But the truncated one stops the gene, it's detected by the cell, and the protein is not made. And there will be no protein, quite simply. And for a number of years, we've wondered whether patients with a deletion, a missing fragment of their chromosome, had the same symptoms, the same phenotype as other patients that had missing letters in their nucleotides. Today, we know that they have the same phenotype. Of course, each person has their own symptoms that are different to other patients, but there is no big variation between their genetic codes. This gene was described for the first time in 2011 by a team that had 6 patients. So they all had gene deletion, and these patients had intellectual disorders, behavioral disorders, obesity or weight problems, and one of them had epilepsy. So 2011, that's not a long time ago, but since 2011, we've been lucky enough to see a lot of papers published, clinical papers or molecular papers. That has enabled us to improve our knowledge, increase our knowledge on these patients that have these random variations. Now, in 2019, when we began here in Rouen at the hospital to look at this syndrome, there were only 14 patients covered in the medical studies. Only 14 had been described. So we only had data for 14 patients. And we already knew then that these patients had neurodevelopmental disorders, problems learning to speak and walk. They were mentally retarded, they had behavioral issues, they had weight issues, epilepsy for some of them, and sometimes even problems with the brain in MRI scans, and many, many things. But in 2019, even though we already had some idea of the phenotype, the syndrome was not very well described. We didn't have good data and not very well phenotyped. And on a clinical and molecular level, we had uncertainties. And that's why we wanted to focus on this disorder. And the main objective was to try and better describe the phenotype, to try and update the data, try and identify new symptoms maybe even, and take a look at whether there was a link between the types of genetic variation, the types of modification on the nucleotides, and the phenotype. And to do that, we set up a team, and they worked for several years. We got in touch with professionals from all around the world, but mainly in France, Belgium, Luxembourg, and the US, and we included 14 patients, as many girls as boys, with a median age of 11. I'll now talk to you about the phenotype that we have come to know in 2025. That's the fruit of all of this data. But what you need to remember is that antenatal, during pregnancy, there are no specific symptoms linked to this disorder, to this syndrome. Of course, there can be malformations, there It can be maternal symptoms, but they aren't necessarily linked to this problem. And today we don't know of any symptoms that mothers-to-be may have. But when at birth, there are no specific problems with growth. Babies are born at full term. They have normal sizes and weights, but 15% of the patients have hypotonia at birth, so restricted movement. And in 100% of cases, these patients go on to have problems walking and speaking in relation to other children, and specifically in speaking. But before that, there are motor problems, so they find it hard to sit up. They do that at 11 months instead of 9, and they walk more like at 18 months rather than 12. And those motor problems also can be seen in the finer motor problems. And then there's that general hypotonia that some patients see for several years. Now, on a language level, there is a delay. First words are said at roughly 2.5 years, and first sentences at 5 years. That can vary. Some patients find it harder than others, but we have these syntax and phonological problems in most patients, and we'll be going into that a little more later. But this neurodevelopmental disorder then develops into cognitive problems in most cases, and they are very variable. Patients sometimes don't have too many problems and others much more. Some patients who don't have mental retardation, according to the international norms, may have learning difficulties. So, for instance, all of the dyslexia, dyspraxia, etc. But there are also visual and attention deficits, and they can also impact the problems, the cognitive problems already present. And most patients need special sessions with all kinds of practitioners to help them. And schooling needs to be adapted, but that can be different from one patient to another. Some only need an extra adult standing with them and helping them a little bit in a normal classroom, but other patients might need to have to go to special needs classes of different kinds. So care can vary a lot, and it depends on the patient's symptoms. Most of the time they have pediatricians following them, sometimes endocrinologists, psychiatrists, ophthalmologists, geneticians, at least once for the diagnosis. And then they have a paramedical care pathway that can evolve with time and according to the needs of the child. Now, as far as behavior goes, we have noticed that the disorders are constant, but can take the form of two different types. Some patients have classic autistic problems. Others have partly autistic problems without being completely on the spectrum. There are intolerance to frustration, sometimes aggressiveness, which can be auto or hetero, sometimes attention deficit, anxiety, hyperactivity, etc. Now, all of these disorders can develop further in life, and those are things that we don't have enough information on. What impacts that change, and they can either be on their own or together, conglomerate. And usually it does have an impact on the patient and on the family. Now, on a neurological level, some patients have epilepsy. We'll be talking about that this afternoon, but we have different kinds of epilepsy. There's no specific kinds of seizures according to what we can see. The severity varies. Mostly it's an epilepsy that starts very early on in the neonatal or early years, and depends on the kind of seizure, what kind of treatment it has. So treatment does normally work, but that depends on the patient.
As far as the neurological issues are concerned, there is no regression, there is no loss of intellectual power or motor capacities. However, this data is limited. and that is something we have not yet observed. No muscle disorder either associated to the gene. However, many patients will show modal hypotonia with coordination disorders. They can be clumsy or they have balance issues. Brain MRIs have been carried out on a number of patients. In 28% of the cases there were abnormalities. However, the abnormalities were not specifically related to the syndrome. We could not find any symptoms in the patients who showed these abnormalities. And most of the time the abnormalities were not really serious and did not warrant subsequent monitoring. So no specific image was identified for this particular disease. Regarding eating habits, many patients, 60% of the patients were overweight or even obese, but this may vary in time, and the patients sometimes will change their weight curve depending on the age, but when they put on weight they did so very early on, in average at 3.5 years of age, and that's very important. This is something that we're going to look into later. And besides the fact that they were overweight or obese, they also had behavioral disorders such as difficulty managing society feeling, hyperphagia. Children who eat very fast are definitely obsessed by food. In a more infrequent way, we found the people with the opposite phenotype, people who had trouble putting on weight, initially very early on in life with troubles sucking on the bottle, or they had to undergo a hypercaloric diet in the early postnatal period. This is rare data, but it can happen. Then we have eye-related disorders, 30% of the cases, not serious, refraction, strabism, also sleep disorders, 33%, which can be have an impact on the patient's quality of life and the family's quality of life. Children who will not go to sleep will wake up at night, and sometimes in the background there may be endocrine disorders such as thyroiditis, microcephaly, which has been described but doesn't really happen very often. We then went on to ask ourselves, people with this syndrome, could they have also show signs in the face that might evoke the MYT1L syndrome because they had developmental retardations, as happens in other diseases. So we worked on the face-to-gene software. It's a machine learning software that uses the photographs published in the literature in order to give us a typical face where we can find signs that are quite evocative of the disease. And these signs can be found in some patients. We have eyes that are sunken and almond-shaped, and the nostrils can be tilted forward, and sometimes there are signs that can be related to the obesity. Now, the symptoms are not constant and consistent for all the patients. Sometimes you will observe them in different syndromes. It's very difficult to make a diagnosis when you see the child for the first time for neurodevelopmental retardation. It's very difficult to think about that syndrome first, only based on the signs seen in the face. We then went on to look at the molecular changes, nucleotide changes in the gene. There are two types of variations. There are protein truncating variations and missense variations. Now, in most cases, the missense variations are the ones that change the protein shape. These variations are clustered. They will cluster together in a very specific place, the second and third domain in zinc finger domains, and some patients show variations that can be found outside of those domains, but that's rare. Whereas the truncating variations that break the gene reading and will lead to the absence of some proteins, they can be found all along the protein. We wondered whether the patients who showed missense variations clustered in the middle of the zinc finger domain had the same phenotype as other patients who had truncating variations. We compared a number of clinical criteria and we could find no difference. The patients had suffered from the same symptoms. There was no phenotype-related variation, no genotype-phenotype variation between the two types of mutations. The assumption, the hypothesis could be that missense variations that change the conformation may be at the origin of a protein. Protein is there, but it's not active. And this has the same consequences as if the protein were missing altogether. Then we looked at the patient's phenotype and compared them when people had a missense variation outside of the zinc finger domain. Unfortunately, we only had 3 such patients, and we could not make any show any difference. So to conclude on the phenotype, the patients showing MYT1L syndrome have neurodevelopmental retardation issues, sometimes psychiatric disorders, sometimes sleep disorders, sometimes eating disorders. They eat fast or they put on weight or become obese. Sometimes they have morphological specificity and behavioral disorders. To conclude, I would like to cover the clinical research associated with the MYT1L syndrome. The fundamental research will be covered later by other speakers. The MYT1L syndrome is a rare disease. The European definition is the following. It's a disease that will affect less than 1 person on 10,000. We think we have like 70 patients in France affected by the disorder. We don't have a database that will help us list all the patients, both in France and abroad. Maybe 1,000 patients across the world, but again, we don't have enough data. As you probably have surmised, this syndrome is a rare disease and we are facing many challenges. First challenge, because it is a rare disease, the patient cohorts are very limited, very few patients in France and abroad, and therefore biological clinical data is limited. If we want to acquire more knowledge, more in-depth knowledge, we have to organize national and international cohorts. This is the first challenge, how to recruit patients from across the world. Second challenge, the data that can be gathered anywhere is unnecessarily comparable because every country has standards regarding overweight and obese patients. The standards are different. It's very difficult to compare the data. This is essential. We need to find a way to perform rigorous scientific studies so that we can interpret the data with the aim of being able to improve the knowledge, but also improve the management of the patients. We can split MYT1L syndrome research in 3 stages. It would be the same for other syndromes. Phenotyping first. Phenotyping means characterizing everything that can be described in terms of symptoms, clinically, but also molecular, for molecular purposes, variations and symptoms. Second stage, we need to find a model, understand the biological models, the cellular models, the animal models, and understand what pathways go awry and when. We focused in Rouen on stage number 1, phenotyping, and stages 2 and 3 are left to the German and American colleagues. Stage 3 is the one we're all pointing towards, how to find therapeutic targets that are specific to the syndrome. So why is it important to characterize at best the phenotype and find the symptoms? Well, because we need to improve the way we attend to those patients on a daily basis. This syndrome is extremely rare, and therefore most healthcare professionals who attend to those children are not familiar with the syndrome. Maybe they haven't even heard about it. The disease was described recently for the first time. It's essential that we train healthcare professionals on this disease and that we come up with follow-up recommendations both for you and for the healthcare professionals. Secondly, based on the clinical characterization, we need to try and understand the underlying biological laws and understand the natural course of the disease so that we can anticipate on some symptoms that could appear later in time, later in life. And also, if we can characterize the symptoms in a more efficient way, then we will be able to describe assessment criteria looking for future therapies we will know exactly what we are looking for, what we're trying to regulate once therapies are available. For instance, if we want to use therapy against epileptic, what is it that we want to control? No more seizures or no more seizures on the electroencephalogram? Do we want to decrease food obsession for food? And it's very important that we understand what exactly we are looking for so that we can adjust the therapies. So we conduct studies, observational studies, interventional studies that may have a bigger impact. We ask people to fill in questionnaires. We ask patients to come back to the center to do a checkup, to collect real-life data so that we can have enough data at a given time, or if we want to go over the natural course of the disease in a more exhaustive way. In this syndrome, there are different issues which require that we perform clinical research. And in Rouen, we focused on two subjects, obesity and eating disorders. And this will be covered by my colleagues later during the meeting. We will show you some slides with the questionnaire that we sent to a number of French-speaking families. And then the second research project on Rouen is regards the neurodevelopmental disorder. The project was started in February. It went on for 2 months. I will explain. And the goal was to improve knowledge in 3 orientations: language, prosody, voice, speech, and cognition and behavior. The goal was to acquire a better understanding of the symptoms, to try and maybe identify specific profiles so that we can adjust the way we manage those patients, and maybe we can find other therapies. For this project, we asked the French-speaking families, because everything was conducted in French, so we had to limit ourselves to French-speaking families, and unfortunately no one from abroad. We asked them to come to the university, the teaching hospital in Rouen for a couple of days to perform 4 interviews, assessment, 1 genetic assessment in order to go over the history, previous therapies, the patient's history, how the patient was managed. And then depending on the patients, we conducted a oral speech assessment. This was done by Constance and Laura, and they will introduce their work this afternoon. Prosody was also assessed, how the patient speaks. And finally, Cécile and Sophie and Hugo looked at the sleeping patterns, and they will deliver a presentation this afternoon. Now, the protocol lasted 1 day, and thanks to the association, we had funding to provide transportation and accommodation for the families, and we saw 1 or 2 patients per day for 2 months. 22 families came to the teaching hospital. We were very lucky. We don't have the results yet. This will be the project for this year when we interpret the results. And just to close, there's another way to gather data, and that's what Valérie was saying earlier. The databases that will be filled in by the families. 2 databases that were in contact with, the French one, Genida, and the American Simon Search Drive. Initially, it was dedicated to research on autism, and the Gemila platform was started by the Strasbourg University. The families send the data based on questionnaire prepared by doctors, and we also worked to adjust the questionnaires for the MYT1L syndrome. And more recently, we restarted the work because we wanted the platform to be fed by the families. And this is international recruitment. It's accessible in many different languages, so the questionnaire can be filled in by many different people speaking different languages. And therefore, we gather results which will be then correlated to the results we obtained with our own studies that we conducted here in France, in Rouen. That's a slide on the Simons Foundation in the States. They offer recommendations for patient management and for the families. There are mini guides for each syndrome, and based on the questionnaire, we gather data from the families, and then we can offer to the family and scientific community all the results we have gathered. I will close with this slide. I think we're all holding hands around the MYT1L syndrome. Clinicians, biological researchers, basic research scientists, and we're all connected to the patients, to their families. We want to work together. We want to improve our knowledge so that we can subsequently improve the patient's daily life and their family's quality of life as well. I would like to thank all the patients, their families, all the families who came to participate in the research project. They filled in questionnaires, they filled in many questionnaires. The association, because we now have been in connection for many years, all the co-workers, doctors, scientists, researchers who worked with us, and the program unit that has been coordinating the projects, the med school that is hosting our work, and all the professionals who worked on this project on a daily basis. Thank you very much.
All right. Can you all hear me? Hello. Good morning, everyone. Also from my side, it's a really extreme pleasure and honor to be here, and I'm absolutely grateful for Valérie. I think she just left the room, for really giving so much energy, love, and heart to this foundation and to make this all a reality, and also for Juliette for pushing this field from the very beginning. And we're very fortunate to have interacted also now over the past few years, and I hope this will intensify. And it's obviously an amazing honor and pleasure to meet all of you, and I hope we can interact in the afternoon and speak a little bit, and so you can hear what we are doing, and we would more than love to hear what you're doing, what are the most important questions that you have, so that we from the basic research side can try to help answer those questions with the utmost importance. As you've heard from the nice introduction from Valérie and Juliette, there's not one MYT1-like patient. We have a tremendous amount of amazing individuals that make up this community, and we try to understand and help everyone a little bit. And also we have an amazing team of partners in Heidelberg that try to solve this puzzle and try to piece it together from a also biological and scientific side. And along with me here in Rouen is Maya Hempel, who you see here on the picture and who's just walking here. So she's our partner in the genetic polyclinic in Heidelberg that very closely is also interested in anything that's related to MYT1-like. And also here is a PhD student from my lab, Jule, also here in the first row. So if you have a chance to interact with her, she's really also studying the molecular basis of MYT1-like syndrome. So our research really is basically focused on a very simple questions. And that is also what was introduced by Juliette, that all of our cells have this amazing capacity to form all cells of the body. And these cells, as you know, are stem cells, and they can form, obviously they can form the brain cells in our body, they can form the muscle cells in our body, and all the other cells. And in order to do so, all those cells have within their nucleus the genetic makeup or the blueprint or recipe book, as Juliette mentioned, that allows basically all these instructions to be read and to make the cells what they're supposed to do. And in order to do so, it is very important that those instructions are read correctly. And for this, there's what we call transcription factors, and those are genetic switches, proteins that go into this recipe book, that's encyclopedia, and finds the right pages, turns them up, and makes sure those are only read. And normally that happens very nicely. Otherwise, the wonder of life, and we all wouldn't be sitting here because there's so many pages that have to be read accurately. However, sometimes this process can also go wrong, and it has to be tightly controlled. And one such very simple image, if you think about it, is that the book of life has instructions for all cells in our body, right? The brain cells that are making us think, that making us feel emotions. But there's also the instructions for the other cell types. And those have to be obviously shut down. And one of our basic findings was that the transcription factor, so MYT1-like, the gene that we are all very interested in, one of its function is to shut down genes, to basically make sure those pages are not opened and read and potentially cause the wrong recipe book to be cooked or the wrong meal to be cooked. This is obviously very simplified, but again, just in a very simplified image, thinking about this, we believe that another analogy to this orchestrator, MYT1L is like a guardian, like a temple guardian that prevents unwanted instructions to be read. And we have studied that for a long time in different contexts, and only recently have also been started to study that in disease, because we have become aware of the work from Juliette and others that there is obviously a potential link between variants in this gene and disease cause. And together with actually very almost simultaneously with the team of Joe Dougherty in the US, we also studied that both in a mouse model and in a human model. And I just want to give you a little bit of an explanation what we do, because it might seem very abstract for some of you. So we want to study what happens if this, what we think is a guardian, is lost in cells. And there are many ways to do so. And obviously, Juliette mentioned where this gene plays a role is in our brain cells. And we cannot easily get brain cells from patients, as you can imagine. But we have now in biological research find ways to generate brain cells in the culture dish, yes. And what we can do in that is, in a very simplified manner, we can take the cells that can do everything, those stem cells, and using genetic tools, such as many of you have heard, this genetic scissors, and change genes. In this case, we made changes or variants that mimic those changes that are also found in patients. And once we do so, we can turn those cells into the brain cells in the culture dish. So this is really a very simplified cartoon. This is a process that takes several weeks and a lot of skills in the lab to achieve. But what it will give you in the matter of a few weeks is this beautiful looking human brain cells in the culture dish, in which we can now study what is happening when this gene MYT1L is changed. And really, I don't go into too much detail, but I just want to give you some of the concepts of what we have found. And one of those is that we started to study the function of those brain cells in the culture dish. And again, just for you to understand how this looks, this is one of those plates which we use to grow these human brain cells on in the lab. And as you can see, there's 48 of these little compartments in which we can grow in the cells, and we can monitor there now the electric activity. As you know, one of the key features of brain cells is to communicate with each other and to exchange electric signals using chemical synapses. And this is how this looks in real life. You have now these electrodes, and on top you see hundreds of thousands of human neurons that kind of lie on that chip that we use in the dish, in the laboratory. And this can pick up the electric activity by monitoring live what those neurons are doing. And so we can look at, are they active, are they not active, how active are they? And I just show you now a little movie how we compare now cells that have normal levels of MYT1L on the left, and how that looks once we have introduced the variants that are also found in patients on the right here, and shown in teal. So each and every of those light sparks that you will see is the activity of hundreds of thousands of neurons that we can monitor live. Again, human cells in which we change now this protein. And it is always remarkable for us in the lab to really see this process live, because we can now see neurons, human brain cells live at work. And I think it's also very clear to you that there's a striking difference between those two conditions. So once we have less of this variant of MYT1L, once we have this variant that is disease associated, we can see a severe hyperactivity. We sometimes refer to that like a little thunderstorm in the culture dish, because there's too much action going on, what a normal neuron would do. And that might maybe relate to some of the experiences you have also with your children. We also then try to translate those findings from this culture dish to a mouse model. And the reason why we do this, because here we can study more complex processes, how the brain develops. Is there anatomical changes? We heard about that there might be micro and macroencephaly is reported in patients. And those features we can also see and recapitulate. But what was very important to us is that this also allows us now to study behavior. And one very simple assay how to look at behavior in mice is just to look at them and see how they behave in an unperturbed situation. And if you now, for example, look at a mouse that has normal levels of MYT1-like, this would run around in the box and just explore the box and, however, stay a little bit to the side, is a little bit afraid and not too active. When we now look at mice in which we have artificially also changed this gene MYT1-like, we can see that they are hyperactive. They run around much more than their control mice, and they have almost 70% increased activity, which is also maybe features that can some of you relate to seen in some of your children. I don't go into much more of those details. We did also, and so did the lab of Joe Doherty, some more tests and assays. One thing I just want to share also with you, however, what we observed is when we looked at the social interaction of those mice, we also observed there was decreased social interest in the mice that had decreased levels of MYT1-like, suggesting really that complex behaviors such as social interaction are changed really just molecularly caused by removing this what we call neuronal safeguard. Really for the first time showing that this could be the cause of the symptoms that we see also translated in patients. So with this knowledge in mind, just again, as a very high level overview of what we observe, because it's all published, and I'm more than happy to tell you a little bit more about the details in the coffee break. What we saw is basically two things. One of the effects when we have a variant of MYT1-like that is associated with disease is that neurons are less good in becoming neurons. So there is a neurodevelopmental phenotype also on the molecular level. And the other phenotype that for us at least was very surprising is that even once the neurons have formed, so the development has occurred, there was a hyperactivity of those cells. And that for us is very striking because there might be phenotypes that are not only occurring in development, but there are maybe phenotypes or symptoms that are later in life that could be targeted with therapeutic intervention. And one such mechanism we tried to tackle because of this, what I told you beforehand, observed hyperactivity. We also could relate it back to a specific potential molecular mechanism because we saw that some channels that are important to make neurons active are increased in those cells. And that is very nice because this allows to potentially use drugs that are used in other diseases such as epilepsy to reduce such activity, to block those channels and decrease hyperactivity. And this is exactly what we tried also first in our mouse models. And as you can see, this is again just now the mouse that has a variant of MYT1-like that is hyperactive. And when we acutely applied this drug, lamotrigine, that is an approved anti-epileptic drug, to the mice, we could also reduce this observed hyperactivity. Also, what is not published, but what we have shown now is that we can also increase the decreased social interaction once we apply this drug acutely. Importantly, we now wanted to take that back, at least on the molecular level, to the patients. Because what I've showed you is all so far only in artificially generated mouse or cell models in which we introduced variants of the gene that are associated with disease. So due to our research and the really nice network of patients that we have been really fortunate to interact, we also now we're taking a different approach. And that is a very, exciting approach in biology in which we can now take the blood of patients, turn this blood into this cells that have this fantastic ability to become any type of cell in the body, which is a pluripotent stem cell. And in this case, turn it obviously into a neuron, which is the cell type that we are mostly interested in because it is affected by MYT1-like syndrome. So just again, also maybe catching back on what Juliette said, one of the questions that we were really interested in was the idea that there are different variants occurring and reported in patients with MYT1-like syndrome. And one is what Juliette also said, some letters are changed, which is missense mutations. So just a single part of this huge protein has one tiny change. while the other big family of changes that occur in patients are changes that lead the protein to be either truncated or completely degraded. And these two, as Juliette said, seem to clinically not have a significant difference. And we also tried to understand this now in actual patient cells. And we were very fortunate, as I told you, to really have now also a nice interaction and cohort of patients that are also excited about this basic research and try to use this knowledge to further our understanding of the disease. And we first wanted to test if now in cells that we get from the healthy family members compared to the children, we see any changes that we observed in this artificially engineered cells. And as you can see, once we test this in cells that really have this loss or decreased level of MYT1, we see the same hyperactivity that we observed in the engineered cells, which makes sense, but it's also very nice to confirm in actual patient cells. And importantly, also those patient cells, really important to state, in culture react to the acute drug treatment with lamotrigine. And then for us, the very important experiment was, as you can see here for Jule, performing those experiments in the lab to test if now this other big family of changes or variants, which is this one changed letter, could have similar effects. Again, when we compared healthy family member cells to cells in which a change in one of those letters occurred, we observed the same hyperactivity, which is in line with the clinical features that Juliette mentioned. And also here, we can, with the acute application of this drug, reduce this hyperactivity. So it's very important to state that these findings are only in preclinical mouse and human cell culture models. We don't know if this has any real-world implications for the patients and the families. There are now several patients in Germany that started to take this medication in collaboration with their treating physicians. However, we have both mixed results. While some anecdotal reports say there is positive effects, several patients also actually stopped the drug because there have been adverse effects. So it's for sure no magic bullet that we have found here. But we're really trying hard to better understand if this could be a treatment for maybe some patients, or understand why in some patients it works while in others it doesn't. So if you or any of your loved ones in your network have taken this medication or are thinking about taking this medication, I would really urge you to get in touch with us, because we need to collect this data to make an informed decision, to make obviously also enable us to give recommendations to families that contact us, because at the moment we are the basic researchers that want to understand this in the culture dish, but we need you to make clinical impact on that. So with this, actually it is important to state there's many open questions that we are also in the lab trying to understand. And I just have some highlights here, and I'm more, as I mentioned, than excited to interact with you to hear what you think is the most exciting questions. So we want to understand the differences between patients now on the molecular level in more detail, to see if what Juliette is observing in the clinic also holds true really in the culture dish. Also, we want to connect these molecular features with the developmental trajectory, so how the kids and patients develop over time. And this is why these meetings are so important for us as basic researchers, also to see children that are of different age, that have different experiences and different developmental trajectories, and learn from that. So it would be really great to connect with every one of you, basically. As I mentioned, the clinical effect of lamotrigine is for us very important, because we think there might be usefulness, but it's, as I said, definitely not a magic bullet. And last but not least, we're also interested in developing additional strategies to potentially have a therapeutic impact in the disease. And Joe Dougherty's team is also following approaches, and it will be very exciting to connect also with researchers in that area. Just one experiment that I want to show why we think there might be also other strategies that could be really more of a therapeutic approach is that, again, coming back to this hyperactivity phenotype, just a different visualization of that is when you have, in this case now, mouse cells that have normal levels of this, what we call neural safeguard, compared to no, you see this hyperactivity. And when in cells, again, in cultured neurons, in mouse, in vitro, we now apply using, let's say, an in the dish gene therapeutic approach by bringing back this transcription factor. In culture, we can also reduce this hyperactivity. At least suggesting as a proof of principle that regulating the levels of these factors by any means, for example, using viral vectors or antisense oligonucleotides, as Joe Dougherty's team is pushing, could really be a therapeutic future avenue. So with this, I have to thank, obviously, the amazing team that stands behind the work. Yule is here, but there have been some really incredibly talented and dedicated PhD students that worked on this project. Bettina, Jana, Bryce, and with the help of Tim and Sarah in the lab. Plus, obviously, the interaction that we have with our amazing clinical partners. You see their faces and names here, most remarkably, Maya Hempel, that is on this journey with us together. And last but not least, obviously, you and your input and feedback, and we really would like to get in much closer interaction with the families and patients to really help make an impact and make a difference. And with this, I'm at the end. This is the family day we organized last year in October in Heidelberg, and it was an amazing experience. It's great to see some faces from that meeting back here, and I hope we can reconnect many times more in the future. And with this, thank you, and I don't know if there's time for questions. If not, just find me later.
Okay. Hello, bonjour.
So good afternoon. Good afternoon. I'm sorry. Thank you very much for giving me the opportunity to speak. I'm going to try and speak French, and I apologize for two reasons. The first reason is I am not used to speaking French, and I'm not very presentable because my luggage was stuck somehow in Montreal. And so I just had to buy an outfit to be decent. Also, I learned French 20 years ago in Belgium, so maybe I have a hint of a Belgian accent, and I apologize for that as well. I started being interested in that topic with my friend, Dr. Constantino, and his patient, Jake. Jake has many challenges. He had a hard time learning. He had also motor disabilities, and back in those days, we didn't know why. But at a time in his life, gene sequencing became available, as Juliette explained, and we realized that he had a mutation of MYT1L gene. I'm sure you know Legos. Genome is just like a set of Legos or Lego. You have 20,000 blocks, and all genes is just one of those 20,000. With Lego, you can do many things. You can design many things by assembling them the way you want. And the brain is maybe like that car. Well, my presentation is just moving along too quickly. So the brain cells are like wheels, for example, of those red blocks. And those red blocks, you can find them in the brain, in the liver. And the genome just contains all those different colors. We realized in Jake that one letter in his instruction book or booklet, if you will, was just changed so that there's this funny looking block. And that's maybe because of this funny looking block that he has so many challenges. Now, you see that if there's one blue block missing on that car, is not too bad. However, if the wheels are missing, then the little car will look like a car, but won't be able to move. So we wanted to understand what are the consequences of having this funny-looking red block. Also, there are limitations and situations when there are translation issues, what we call nonsense or missense. And is it okay in French? Well, I can switch to English if you prefer. So, Juliette explained very nicely the problems we face with our patients. She talked about different areas where we see challenges, motor skills, motor delay, speech delay, behavioral changes, and changes in the brain development. So, we decided to start our program with the Jacob Gene Fund and with a pilot study. We built a mouse model and a cell model in order to reproduce Jake's variants. The first thing we learned is that one of his cells had a nonsense mutation. And then we also had a mouse model, as explained before. They are similar. I'm sorry, it's very hard to translate his French. I apologize for saying that, but it's very difficult. So we wanted to use those mice to understand what's the impact on the neighboring genes. We added a mutation that's reasonably similar, and we had individual cells that all carry the same mutation, and we simulated what happens in the brain. So what can we learn from that model, and what can we do? My colleague had a look at those cells because a number of things can be learned from studying cells. Together, we started a project between 2020 and 2025, and we had 3 research questions. The first question, we wanted to know what are the consequences on cells and brain circuits of having a bit less of the funny Lego piece. I work with Susan Maloney. She's an expert in mouse models. She's very good at understanding how they move, what are their learning abilities, what are their behaviors, and how we can learn from them. Now, the first thing that's rather easy to spot is when mice are hyperactive, and we compare brothers and sisters, if you will, from the same litter of mice. And so, on one side, you have 2 mice from the same litter, and on the other hand, you have mice with a simulation of a MYT1L syndrome. Now, if you look at that mouse, you see that there's a pretty repetitive behavior. And we organize Olympic Games for those mice. We ask or instruct them to climb that pole or walk on a grate. But quite often they're faced with challenges. They have a hard time doing what they're instructed to do.
And I think it got the students very excited. So we now have 4 different people in the lab working on MYT1L. We have a physician scientist who's joined us, who's going to talk next and tell us about that. So we're very excited about our continued studies going forward. So I want to thank my team. I've told you about Chris Kroll and Susan. I think the people who really led the work on the former population of students who led the work were Alan Yen, Jiayang Chen, Simona Serafinoska, and of course the group that are working now on it, like Allison and Colin and Mary. And a new student just joined who's all excited to work on MYT1L. So I want to thank the lab and thank you for your attention. Could show how much it liked social behavior by having these 2 little holes, and it could kind of poke a hole, and if it did, it would open a little door. He's going to poke right there, and the door goes up, and he gets to sniff his little friend for 10 seconds. So it's a way of sort of quantifying how social the mice were. And generally, they were fairly social, so that was pretty normal in the mice. Maybe there's some subtle differences in the males. We also wanted to look at learning, and we looked at things. There are a lot of classic tasks in mice to look at how well do they remember their locations, how well do they remember a shock, things like that. And the mice seemed pretty normal on their memory for locations, but it wasn't very sensitive to learning. So we tried to develop something that was a little bit more challenging for the mice. And so my friend Lex Kravitz invented this little device, a little toy that goes in their home cage where they can learn to play a game. In this game, they poke their nose in a hole here, and they'll get a little prize. So here's a little mouse doing this. Oh, ça va marcher, non? Well, this video is not working, but what it would show is he pokes his nose in the hole, and a little piece of food drops out, and he excitedly goes to eat it. We can make the game a little harder by having 2 holes. And we make it so that initially the mouse learns that the prize is on the left. And he'll push and he'll push and he'll push and get more and more prizes. And then we will trick the mouse and move it to the right. And then after about 30 tries, we'll trick him again and move it back to the left. And so they have to learn to go back and forth and be flexible and be able to change their behavior as the demands change. And the thought was maybe with MYT1L, they have more challenges in changing their behaviors. So we change kind of every 30 pokes. Oop, the little mouse moves. So this is what the siblings look like, the brothers and sisters. So here's which side is getting the reward. It's either getting it on the right or the left or the right or the left. That's the black line. And here's the behavior of the mouse. They initially don't know what to do. They know they get some rewards. So they try both sides. But over the course of days, they learn, I should switch to the left. I should switch to the right. I should switch to the left. The mice with the MYT1L mutations had a lot more challenges with this. They did eventually learn it, but not ever quite as effectively. They tended to kind of stay a little more in one place and have a little bit of trouble switching back and forth. So we think this is kind of a sensitive measure of that learning ability and repetitive behaviors. We also did some brain imaging, and as reported in some of the patients, there's sometimes a tendency for slightly smaller brains, but grossly these things are pretty normal. They're not missing major structures. It's pretty subtle. So it's mostly building a brain you would expect, just a little bit smaller. And we're starting to try and do this at higher resolution now and actually measure the counts of different kinds of cells in the brain to see if if all the normal cell populations are using these new spatial transcriptomic techniques that I might have to tell you about in 2 or 3 years because we're just starting these experiments. I guess one last, so we were doing all this to be able to understand if the mice have the same challenges, because then it gives us a platform to test lots of therapeutics. There's only one Jake, so Dr. Constantino can only try so many things with Jake, but having the mice, this gives us the opportunity to try lots of different things quickly and see what might work. And so seeing that they have similar challenges, then gives us we can repeat those same assays with different treatments, as Dr. Mall showed you in his experiments. So we're hoping that'll be something we can do. The other thing that's nice about mice is they have much shorter lifespans, so we're able to look at kind of longitudinally how does this affect development. And I think there was some good news there. I mean, to the extent that mice are similar to humans, we see a very similar lifespan in both them and their siblings. It's not very different. So there's because sometimes some neurodevelopmental disorders also come with heart conditions or things that affect later in life. We didn't see any of that here, so that was reassuring. So they have many of the same challenges. Why? Well, we think the smaller brains may be because of a little decision during brain development. There are cells who proliferate and double to make more cells to grow the brain when it's young, and there's other cells that differentiate to make neurons. If the cells switch too early to making neurons, you don't quite grow the brain as much as you would like to, and so we think that might be why the brains are slightly smaller, is an early switch to making neurons. What does the gene normally do during development? MYT1L is a gene who controls the expression of other genes. It can be both a gene turn on some genes, and it can also turn off genes. And so that's a little confusing because going in, we thought it would be one or the other. It turns out it's both. It does, at some genes it turns them on and some it turns them off. So there will still be a lot of work to do there to understand what its targets are and why it plays this strange role. Generally, I think both labs that study this saw that it turned on and off very similar genes, which are genes involved in the maturation of neurons. And I think a broad thing that we're seeing is that just like the neurons are born too early, they kind of rush into being neurons, but then they don't really mature all the way as you track them over time. So it seems like they don't quite get to the mature state in the brain from what we can tell in terms of their gene expression. Chris Kroll, our colleague, saw similar things in cell culture, so we think this is true also in the human neurons, as best as we can tell, in terms of staining with markers of maturation, for example, like this kind of MAP2 staining. So that's where we've gotten so far in the last 5 years. I think it's kind of been a lot of platform building to try and be able to create an opportunity to test different treatments with these different platforms. We're looking forward to working with Dr. Mall in our next round, where he in parallel made a different patient mutation. And so we have basically 2 mutations to study. We're hoping to use those in parallel. And the goal for our next 5 years, if funding for science in the US does not collapse, which is at a fair amount of risk at the moment, is to try and understand a lot more of the missense variants. Those variants where we don't know whether or not they have exactly the same consequence as someone who's lost MYT1L levels. The other thing we want to try and do is use what we've learned about MYT1L gene expression to build better diagnostic tools for those missense variants. Looking in the patient records, it seems about half of the variants are missense, and for those, we don't really know if it's exactly the same syndrome or if it's doing something different. The missense variants, for example, might only activate genes or only turn off genes instead of doing both things. And so that's something we want to study in depth so we can help people get better diagnoses and figure out whether these are similar related disorders or very distinct. And finally, take these platforms we've built and try and screen for medicines that'll address these challenges. We have some high risk, high reward studies also about trying to test variations on gene therapies with RNA therapeutics. Because it seems like if you're making too little of the protein, one thing you might want to try is just driving the creation of more of the protein, simply adding it back. But it's unclear the right time to do that. If MYT1L is mostly needed in early development and you add it back later, maybe it doesn't help so much. So the mouse will let us to tease that out if we can build the right tools to let us turn the genes on and off. And that's an area of active research, as well as trying to develop RNA-based therapeutics that would be translatable from mouse to human. And I have a variety of students in the lab who've gotten very excited about this. I think one of the best things about this was a turn from my lab. I was a fundamental, basic researcher in kind of gene expression in the brain, but working with Jake and Dr. Constantino gave me a chance to still explore that fascinating biology, but also do something that had an impact on patients and connections with families. And I think that meant a lot to us also. We had the first group of families come to St. Louis in the US, some of them who I see here today. And I think that was really enlightening for us and changed how we think about the research we do. So we learned a lot from you guys who are really the experts in the disorder. And I think it got the students very excited. So we now have 4 different people in the lab working on MYT1L. We have a physician scientist who's joined us, who's going to talk next and tell us about that. So we're very excited about our continued studies going forward. So I want to thank my team. I've told you about Chris Kroll and Susan. I think the people who really led the work on the former population of students who led the work were Alan Yen, Jiayang Chen, and Simona Serafinoska, and of course the group that are working now on it, like Allison and Colin and Mary. And a new student just joined who's all excited to work on MYT1L. So I want to thank the lab and thank you for your attention.
Bonjour à tous, je suis ravi d'être parmi vous aujourd'hui et merci pour l'invitation à cette conférence. Et j'ai le plaisir de partager la recherche clinique menée des États-Unis. And that's all the French you'll get from me today. Okay, so let's talk a little bit about MYT1L syndrome and the state of clinical research in the United States. So first of all, yes, I'm a medical geneticist, and I've been collaborating with Joe Dougherty, kind of on the clinical side, trying to connect the lab and the clinic to make whatever discoveries are made in the lab relevant to patient care. And the first thing I want to talk about is, first of all, like why do we want to do clinical studies? And I won't go through the whole list, but first, of course, as Juliette has already mentioned, we want to learn more about the condition and see what's important, see what we can target, and also eventually knowing more about the condition can help us guide medical care. And eventually, and that's what we all want, is have clear guidelines and tools to treat our patients with MYT1L syndrome. I want to use Rett syndrome as an example, not necessarily because it's similar to MYT1L, because it's really not, it's very different, but it's one of these rare disorders that we've known a lot about it for a long time. And we can put it as an example of how we learn more about a disorder, and then how we eventually get to some type of intervention and treatment. And again, just quickly going through this, first, Rett syndrome was first described in 1966. And since then, a lot of things have happened. Of course, number one, defining the disease, what it is, what it's not, creating animal models, establishing a natural history study to learn more about the condition, its course, and eventually finding out what traits are reversible in mice and could potentially be treatable in Rett syndrome, and then testing drugs in mice, and then eventually getting the first FDA-approved drug, trofinetide, in 2023. So again, it was like almost 50 years from the discovery to the end. Hopefully that's not what we're expecting on other genetic disorders, because things hopefully go quicker these days, but this is just to give a comparison of kind of like where we are right now in terms of MYT1L syndrome from the first report of the deletion encompassing MYT1L to the mouse model. And now going specifically to what we've done in the United States from a clinical standpoint, we basically started last year, it was in May 2024, with a MYT1L focus group. So we've met online with a total of 6 families, and with a goal basically of course, making those connections, but also trying to find out what is important for MYT1L families. Again, 6 families is a very small number, but that data could give us a guidance of what's important for some of those families, what are the next steps, and where to go from there. So yeah, we discuss experience, challenges, strengths, priorities. Then, as we've mentioned a couple of times already, in October, with the help of Joe Dougherty and different Winwell families, we had the first scientist family meeting in St. Louis, Missouri. Oh, sorry, didn't update this. So we had a total of 8 in-person participants. What I mean with that, 8 is 8 MYT1L families plus researchers. So it's not 8 in total. We had a lot of online participants, but we weren't sure like how many of those were families versus other interested researchers not. But at the end of this meeting, we sent a survey to different families, and we had 33 respondents. So at least we had 3 families somewhat participate. 33 families participate in this meeting. These are just some results from that survey. Again, it was a very informal survey asking a lot of open questions about priorities for families, or what were the symptoms that families thought were most important to treat or target. And in this graph, I'm just showing out of those 33 respondents, what they consider was the priority number 1 sign or symptom, and priority number 2. Priority number 1 being the blue columns here, and then the orange ones are priority number 2. So as you can see, some of the highest ones are basically related to behavior, mental health, and temper and outbursts. So which again, kind of significantly related. Curiously enough, intellectual disability was mentioned as high priority by at least 25% of respondents, but then was less of a second priority. And again, maybe some important mentions here, like we've been talking a lot about obesity and GI issues, which are still are of course significant too. About 10% mention it as a significant symptom, but also communication was kind of at the same level. There was a very open-ended question too for comments, and I thought the best way to show kind of what the comments were, what their significance were, was like to create this word cloud here about the most frequently mentioned words. And as you can see here, a lot of respondents mentioned outbursts as something of significance, as well as obesity and communication. And again, there's other several things there, but that kind of shows you that at least in those 33 respondents, not necessarily everyone, but a nice number said that outbursts, communication, obesity, behavior also were significant features here that require some type of intervention for their children and loved ones. We also asked them about medications, like what type of medications were they on, and I'm showing you here a graph with some of those medications, and by category, and probably the most frequent ones were ADHD medications. Number 1, because it's one of the most treatable things there is. Like we have good knowledge about how to treat ADHD. But there's also like some other mentions like GI meds. It seems like constipation was something that some families mentioned, at least I think 20 or 25% of families did mention that. And some other like anti-seizure, anti-epilepsy drugs were also mentioned. Of course, clarifying that even though they're in this category, doesn't mean that they were being given for epilepsy, because as we mentioned, like lamotrigine is a med for seizures, but it's also used for other things like mood modulation. So that was the survey that we did, but we were also working with this registry called the Brain Gene Registry. It's a national brain gene registry, like in the United States, where basically it's a registry of patients with variants or changes in what we call brain genes, so genes associated with neurodevelopmental disorders and other brain-related conditions. And within that list, MYT1L is there, so we kind of like want to take advantage of that already established platform in order to get more information about patients with MYT1L. And there they provide, of course, participants can share electronic medical records. There's also a rapid neurobehavioral assessment protocol. So basically, it's a neuropsychological test, but adapted to a teleconsultation, telemedicine. So it's basically done all via telehealth, via Zoom or a similar platform. They share the genetic testing report, and there's also some other health surveys included there. Unfortunately, this registry is only at this time for people living in the U.S., so that's quite unfortunate. So far, we have recruited 22. There are 22 patients enrolled. We have information on 18 of them, and again, there's different types of variants from missense variants to truncating variants, deletions, and some what we call intragenic duplications. And something else that you can see there is that we're also including some patients with variants of uncertain significance. So whenever we're interpreting these results, have to take that into account. But basically, the philosophy of this registry is to gather as much information on anyone with a gene change, even if it's of uncertain significance. And I'll go very quickly through this again because these are very crowded slides. But some of the data mining that we've been doing, we've got some preliminary data comparing like some of the most frequent diagnoses found in the medical health records. Of course, unsurprisingly, lack of normal development is the one that we've found in 100% of the participants. The red, orange bars represent MYT1L patients. The blue ones represent all other patients in that database. And so we decided to kind of compare them a little bit. You can see that, at least in that cohort, the prevalence of things like lack of normal physiological development is much higher compared to the whole general BGR cohort. And some other things that as expected, things like constipation that I've mentioned, obesity, it's at least like 25%, and other GI issues. Some medications that some people that we found that some people had been ordered at least once. Unsurprisingly, a lot of like ADHD medications were there, like guanfacine, at least in 4 of those patients. And these are some preliminary results on the rapid neurobehavioral testing. Again, these are based on a very, very limited number of patients because even though 22 patients have been enrolled, not everybody has completed all the tests. But I think this kind of like tells us a little bit of what we already know about MYT1L, but it maybe tells us a little bit in a quantitative way with numbers. Again, with a very limited number of patients. So, for example, in this graph, I'm showing the result of this specific tool, the Shipley Standard Score, which tests cognitive ability. This curve here just represents the general population. This peak being the average in the general population, the average score, which is 100. This is only like 5 MYT1L patients that did this test. But again, so limited conclusions here. But as you can see, overall the scoring is below average compared to the general population. Here it's another tool used in younger individuals and in people that have maybe like more cognitive delays. And as you can see here that there's also a trend towards everybody being below average and several of them being like below the threshold for what we would consider like the developmental delays. And this is even more crowded. Again, you don't have to go. I'm not going through each one of this, but this is what we call the Vineland tool, which basically looks at adaptive behavior, which is like practical everyday skills. This is like the general score, like the total score, again showing that all patients are below average compared to the general population. And these are like sub-scores, like basically looking at different sub-skills, like for example, like socialization, like daily living skills, like motor skills. And overall, what we're seeing here in this very limited number of patients is that all of them have in general some degree of delay in different subcategories, in different realms of adaptive behavior. So this is where we are right now in the US in terms of clinical studies. What are we envisioning in the future? Of course, I still think that the BGR is a nice tool because it's already there, it's established. So we're kind of like motivating like US patients to get enrolled there. But we, as we're doing today, we are promoting collaborations with other clinical and family groups in the US and abroad. I think one of the end goals is to, at some point, establish a natural history study. Again, working with my university, but also working with parents groups abroad and also the parent groups in the US. So we can eventually know more about MYT1L and what type of interventions could help our patients in the future. And that's all that I have. Thank you very much.